So welcome everyone again to our July Catalyze webinar. This webinar will provide an overview of key elements of early stage drug development to consider when seeking translational funding. So today I'm very happy to introduce today's presenters, Jon Collins and Bryan Baines. Jon Collins is the CSO of Pinnacle Hill and Director of Therapeutic Research Translation at UNC Chapel Hill. And Bryan Baines is Director of Scientific Collaborations and is the liaison to 4 Points Innovation and brings more than 30 years of broad knowledge and business experience in the biopharmaceutical industry. So with that, Jon, I'm happy to turn over today's webinar to you.
Thank you. Thank you, Marissa. Thank you, Patrick. We appreciate it. Bryan and I are happy to be here today. It's a pleasure to be able to talk to people about, about this topic today. This topic today, as Marissa just said, from target to translational funding, the journey from academic discovery to an investable therapeutic. And by default, given the title, this talk is actually going to be geared, very focused on academic investigators. And we hope that those who aren't in the academic community, there's a few gold nuggets in here for you as well. Bryan and our respective roles, me at UNC Chapel Hill, Bryan at Duke, we strive to engage really early academic investigators. And Bryan, you always say this, you always say, what is it? You can't come to me too early, but you can come to me too late, right?
Yep.
So, and part of that is to help define strategy, a path to IP, what's the strategy for IP funding, for follow-on funding, whether it's government and non-government. And, I'll say this now, drug development is expensive, as you'll see. And you will need, funding beyond just government funding at some point, because it's just really expensive and really risky. So hopefully today you'll walk away with some useful information for your respective projects. Jump forward here. So here's an overview of the presentation today. These are some of the topics. There's a lot. Drug development it's a long, complicated process, expensive process. Bryan and I talked a lot about how we wanted to, what do we wanted to cover today. So we kind of cherry picked a few topics which are highlighted here, and a lot of this is based on our experiences engaging faculty, our experiences, our observations, some of the key learnings over our years of experience talking me in pharma and in academia, Bryan, out in various roles, and in particular at Duke. So at the top, begin with the end goal in mind. This is something that, in our experience, for a lot of academic investigators, they tend to kind of build projects from the ground up. And when you're thinking about drug development, it is really, really essential that you think about the end goal at the very beginning of the project. And we'll touch on that in the next slide. It's kind of a change in mindset. What is the product? What do you want to deliver? We'll talk a little bit about drug development cost, failure, and risk. And I know people know this, some of this, probably not all of this, but there's been a real evolution, I'd say, in the last 5 to 10 years in terms of how translational funding groups, investors, and pharma companies think about targets, target selection, target validation. We'll touch a little bit on unmet need and medicine proposition. I think at the end, we'll talk a little bit about commercial considerations and translational funding landscape as well. From what we understand in this same forum, there have been prior presentation about the target product profile. This slide, it's unpopulated with information, but it's really to try to remind people this is really, really essential for investigators or researchers to really think about, what is your target product profile? What are you going to market? And hopefully you'll get some reimbursement for that as well. Otherwise, it won't be, a viable product. These topics include, what is your modality? And we'll touch on that a little bit, but I think it's really important that the academic investigators really kind of reach out to people who have the know-how to kind of help them figure out, what is the best modality for my particular project? What is the best indication? How are you going to find the patients that are really going to be the responders for your potential therapeutic? Your mechanism of action, how's that different than everything that's out there? And I won't go through all of these, but down at the bottom, efficacy, safety profile, really, really important. Efficacy in simplistic terms, efficacy and safety profile kind of defines your therapeutic index, and therapeutic index in oncology is very different than therapeutic index in obesity and type 2 diabetes. So this is a living document. We encourage teams to really stop and think about, what is that product you really want to ultimately deliver at the end of the project. And so I really want to touch on some of the risk and cost with drug development. And this I should point out now, these data are taken from the bottom right, 2010. So it's relatively old, but I think it, for the purposes of this talk, it kind of highlights a few points that I'll make. On the left-hand side, on the y-axis, you see probability of success. And on the x-axis, what you see is the various phases of drug development, everything from target validation to clinical development to launch. And if you focus on the blue line, what you'll see is this is the probability of a project moving from one phase to the next phase. So if you pick target validation, there's about 25% probability of success of that project moving to hit ID. And then as you get to hit ID, then, it's above, say, around 75% probability of success of transitioning to the next phases. All the way out to, phase one, it's reasonably consistent. But what you see is a significant drop off in phase two. And phase two is a really, really, as people know, really, really critical stage for a project because it kind of defines your proof of concept for your therapeutic and your, therapeutic hypothesis. And then beyond phase two, the probability increases as you get to launch. If you focus on the red line, what this does is it looks at the probability of success to approval for a given project. And if you look at you see 1.3% under target validation. So there's a 99% chance at target stage that you're going to fail. And then even if you get to phase one, there's still only a 14% chance that you're going to get to approval. So there's a lot of attrition here in drug development. This example here is a monoclonal antibody example. On the right-hand side, what this does is capture the cost by the same x-axis, the different phases of drug development. And I'll just focus on the red line. What you see from target validation up to phase one, it's expensive. It can be up to $50 million. And again, I think at some point for either early stage biotechs or academic investigators, government funding will get you started, but you will eventually need some other translational funding, investment funding, or partnerships with pharma. You will need it because it's really expensive and the attrition is high. If you keep progressing, what you notice is that the cumulative cost per program just continues to rise, but there's a really dramatic increase in the cost for a given project once you enter clinical development. And if you look at the green line, and what this is this accounts for all of the attrition on the left-hand side, these numbers, and these are all numbers. So this is the green line is expected cost per successful program. And again, there's a lot of failure leading up to these. And what you see is, an increase, as you might expect, for the cost per program up to phase one. And then you get this dramatic increase because of phase one, phase two, phase three, and then obviously at launch, as you start generating revenue, it starts to stabilize. But the cost can be $1.2 billion. And if you look at this region here, this is about 70% of the total cost for a drug development project, 60% of your time. And again, at phase one, there's only a 14% chance. So even once you dose a human with your therapeutic, there's only a 14% chance this is going to be successful. So it's expensive and it's really, really risky. So why is there so much attrition at phase two and phase three? And these are some figure that took from a couple of papers here. References are down at the bottom. What you see is phase two failures, 2008, 2010, about 150 drugs, and phase three submission failures, 120 drugs. And what you see on the bottom, these pie charts, is that while safety, is a key reason why drugs fail, it's really efficacy or lack of efficacy or sufficient efficacy or sufficient therapeutic index. And this is what, 10 years old now, it's a decade old. So, a lot of companies and translational funding groups and others have started to kind of rethink how, they think about how to manage the cost, manage the risk for a given therapeutic project. And about this same time, there, as these papers on the left-hand side, these papers on the right-hand side, Matt Nelson, who's now head of genetics at Deerfield, he used to be head of genetics at GSK, and the Broad Institute published a couple of interesting seminal papers in this space. And there are other publications that have come out after these dates. And what they found, they did an analysis and looked at the link between genetic evidence or prior genetic evidence for a given target and the probability of success of getting to a drug. And what they found is kind of summarized at the top here. And I cherry-picked some of the data here in these two on the right-hand side. And what you see here is, you can look at a variety of different target genetic data associations. And OMIM, people are probably familiar with that out of Johns Hopkins. GWAS, I'm sure people are familiar with. And this one here, this RVIS, this residual variation intolerance score. And I was less familiar with that. And it's basically amino acid changes in a given gene versus the genomic average. So it just gives you a feel for, how these residual changes are tolerated. And what you see in general on the x-axis is an odds ratio. And this is only for the druggable genome. They've also got undruggable genome in Matt Nelson's paper. And what you see is increased odds ratio if you look for prior genetic data to support your therapeutic hypothesis for your target. And down at the bottom, it focuses just on clinical development. And I'll just jump to the bottom here, phase 1 to approval. There's about a 2-fold increase in your probability of success of getting to approval if you have prior genetic evidence associating your target to your phenotype or your disease indication. So this has really led to a shift in mindset for translational funding groups, investors, pharma companies. Now they really use this as a key filter. And for those individuals in academic institutions, I'm sure you've seen these RFPs that come out for projects. And I would guess that, at least the ones I see, the majority of those, they will have some requirement for human genetic data or human translational data. And part of that stems from the fact that mouse models and rat models are just not predictive of human biology and pharmacology. So here, in a nutshell, relative to the risk and cost that I just showed you, if you can increase your odds of success by 2-fold, think about what that can do to your cost and your timelines when you're spending $1.2 billion. It can really have a dramatic impact on the economics of drug development. And that's why a lot of external groups have shifted and started to focus on this criteria. And here's some data just to kind of back that up even further. Is on the left-hand side, this is basically top 20 most prescribed drugs and the top 20 grossing drugs. And what you see on the left-hand side is about 53% of these targets, they're supporting genetic evidence. And on the right-hand side, there's 8 out of those 20 are in oncology. And as we all know, oncology genetics is very different than non-oncology genetics. So eliminated those. 2 out of the top 20 for infectious disease, who eliminated those. That leaves about 10. And of those, that's 7 unique mechanisms of action. And out of those, 86% are supported by human genetic evidence. So, there's reason to believe that, trying to get an early read on the link between your target and a potential indication using human genetic data will really increase your odds of getting to approval in a product. Not only, and this is a very recent paper, I encourage people who haven't seen this yet to take a look at it, Nature Reviews Drug Discovery, is that not only can you look at the potential link between your target, your gene, and potential signs for efficacy, you can also look for potential side effects, which it is obviously, the sooner you know those in a drug development program, the sooner you can begin to think about how you're going to mitigate that risk. On the left-hand side, and I should point out now, this is target safety. And for small molecules, and I'm a small molecule guy, so I tend to think about small molecules more than any other modality. But I think a lot of this is true for any modality. These data, these genetic association data are for the target, the gene, not your therapeutic product itself. There could be separate side effects as a result of that particular modality. So yeah, we focus on the potential efficacy, indication, why. And if you dig and look through this, you can actually find potential markers of potential side effects. And again, most of the individuals that I've collaborated with, worked with over the years is, most people early on don't really think about side effects. But the sooner you can identify those, the better off you can begin to, the quicker you can begin to mitigate that risk. Because I can assure you, those individuals in the translational funding space and investors and pharma companies, they will know this. So they're going to start thinking about this. You can look for, and this is in publicly available data sets, you can look for individuals that have loss of function mutations, and you can look at the phenotype of those individuals and kind of tells you a little bit about, are they tolerated, what could happen. If you've got an agonist project or an antagonist project, and you can look at that data, it can give you some early read on what could happen in a human. And this is really useful because it not only does it help you build, and this is oncology, just for example, a link between your target, modulation of your target, and potential phenotype in, for example, this organoid or cell assay, but it can also help you in guiding your selection of assays for secondary pharmacology. And if you know that your side effect is associated with elevated blood pressure or hypotension, then you can begin to monitor for that in your preclinical drug discovery project. So I think we all appreciate that target validation is really, really critical. And really, I think the two key questions you try to answer are shown at the top here. What is the link between my target and the disease process? And will modulation of that target be effective in a defined patient population? And I've listed some of these bullets down at the bottom to think about how do you go about establishing a target disease linkage. And some of this I'm sure people know about, others may not. I don't really know. We've talked a little bit about human genetic data, and human genetic data, you can get coding, loss of function, or gain of function mutations in your gene that you kind of know, okay, yeah, this is going to be a gain of function or loss of function. But there's a lot of human genetic target associations which are in antigenic regions or introns or other regulatory regions. And it's really, really important to kind of dig into what's known about the functional consequences of that, because it kind of tells you the type of pharmacology you might need. So what are those functional consequences? And then that may require experimental validation using some functional genomic studies. And while here we're focused a little bit on human genetic data, I will point out, I think this is equally important. If you've got human proof of concept data, if there's a drug on the market that's got a particular mechanism of action, the biology has been de-risked in humans, so we know the therapeutic hypothesis is valid, but yet you've got a unique approach that, you could get greater efficacy with your approach or less side effects. That counts to me as good, strong human translational data. If you don't have that, are there other, human translational data you could acquire? So I think thinking about how do you build that into your preclinical drug discovery project is really, really important. And if you do that, it just gets people to pay attention to your project once they see, okay, some of the biology has been de-risked in the humanized system. What about your target, the signaling pathway and implications in disease, the interacting partners? Are they genetically linked to your disease or your phenotype or clinically? So the more you can do to try to understand, what's the strength of your therapeutic hypothesis in the context of a human, not a mouse or a rat. People, everybody uses genetic techniques or pharmacological techniques to manipulate the target to kind of show that they've got a desired phenotype. I encourage people to really, and I see a lot of experiments in mice and mouse cell lines, which are great, but in the end, I think that you have to begin to think about, well, okay, how do we begin to test this in the humanized system? And sometimes that may mean you might need to try to get access to patient-derived samples. And again, if you can generate proof of concept data in humanized mouse or using patient-derived samples, it just de-risks the project from the minds of funders, commercial funders for a given project. Bryan, do you have any comments on this here?
No, I totally agree, Jon. And I think that the last comment you made is really correct, that what we're trying to do with a lot of this stuff is we're trying to de-risk it in the eyes of future investors. Because once you start moving out of the academic landscape and government grants and into some of these other funding mechanisms, their thought process is just quite different, right? And they're just trying to de-risk things a lot sooner. So that hockey stick, if you will, they can feel more comfortable about the fact that when they get to phase 2, there's a better chance of success because they're going to have to pour a lot of money into it, right?
Right. Thanks, Bryan. And then, you can read the last two down at the bottom, which are important to think about as well. And then, again, I mentioned I'm a small molecule guy, and but I think these general principles could be applied to any, therapeutic modality. The quality of a tool that you're using, especially in an academic setting, in any setting, but I think people in commercial settings, pharma, mid-stage, late-stage biotechs actually think about this by default. So really think about the quality of the tool you're using for your in vitro and in vivo POC. Understand, what the properties are. And the reason why this is important, because this is your proof of concept data. It gives you a mechanism to actually establish a link to disease biology, and then begin to look at, okay, if it's a small molecule, are there some chemotype related, effects, cell toxicity or in vivo toxicity? So I've listed some properties down at the bottom for a small molecule. Binding, adequate binding, less than, say, 100 nanomolar. You understand the pharmacology. You've got all the data to back that up. Selectivity, this is something that I find that a lot of people just ignore. They just think about their target of interest and don't think about family selectivity. And there could be some liabilities or reasons why you might want, dual activity. There could be reasons why you don't want. You want to have selectivity over other family members. And when I say selectivity, in my mind, we tend to think 10-fold at least, but ideally you want to have 100-fold selectivity over other family members. And part of that, so you can really begin to interpret the readouts from your in vitro and in vivo studies. Solubility for small molecules is really important. There are a lot of non-specific effects that compounds that aggregate or insoluble, that it just makes it complicated to interpret the results. And people in the commercial world or, the funding world actually think about this all the time. What is, are these compounds in solution? Are they aggregating? Are the effects specific? Are they non-specific? Is it because of another target? Is it because of your target of interest? For intracellular targets, demonstrating, we know our compounds get into cells. They're permeable. There's no efflux, PGP or otherwise. Log P is at octanol water partitioning. There are a lot of algorithms out there. Swiss ADME is one that I use sometimes for generating calculated log P. And the reason why this is important is it's well established now that the compounds that have high C log P, above, say, 5-ish or so, they're promiscuous, and there's a really, really increased risk of toxicity. So you want to, if you got hits that are in that 5 C log P of 5, you want to kind of figure out how you're going to drive down the C log P. Is not the tools, make sure your tool's not chemically reactive. I see a lot of molecules from academic investigators where there's this embedded reactive group that they don't really appreciate. So just make sure you've done your due diligence there, looking for those things, unless it's actually your desired mechanism of action, in which case that's a very different proposition, and those need to be well characterized. In my experience, at least in the chemical biology world, that a structurally related inactive analog can be a great control for a small molecule project. Okay, so this is a slide, Bryan and I thought would be useful for people with it. Now, hopefully, everyone, the attendees here, the participants, really understand these 4 pillars of pharmacological tool validation, and the original papers down at the bottom. If I had to cherry pick one slide that people remember it's this one right here. These are a lot of key questions in these embedded in these pillars. Exposure at site of action. Is my therapeutic getting to the site of action? Is it engaging the target? Is it eliciting functional pharmacology? And is it that delivering a disease-relevant phenotype? And on the left-hand side, I've kind of listed some of the ways you can measure these. And again, small molecule focused. Apologize to those with antibodies or gene therapies or cell therapies. Exposure at the site of action, if you can look for in your assays when you're optimizing your compounds, biochemical and whole cell activities correlation. There are experiments you can do to use an LC-MS to measure the concentration, intracellular concentration of your compound. Target engagement. A lot of people, I strive to get some kind of functional probe or endpoint that allows you to get a handle on, is my molecule getting to the target of interest? And if you got an enzyme inhibitor, for example, you look for buildup of substrate or loss of product in that cell system. Proteomics methods sets us a cell-based thermal shift method for looking for confirmation of target engagement, but it can be challenging. And what a lot of people do is they'll default to just functional pharmacology. If you got a kinase inhibitor, you'll look at a downstream phosphorylate kinase. If you got a demethylase inhibitor, or you look for changes in methylated histone or levels of secreted protein, or whatever the case may be. But I think when you're thinking about what do you measure, I think, to really convince yourself that your therapeutic is getting to the right target and doing what it's supposed to, you want to pick a biomarker that's proximal to your target, your site of target engagement. Down at the bottom, desired phenotypic perturbation, and just here's one example: bronchorelaxation of airway tissue. That's just one measure, one example. But it's ideal to have a high degree of confidence in, obviously, the clinical translatability and hopefully disease relevance. So building their preclinical assays, and the one on the left is probably a little less relevant for this presentation, but I put it in here anyway, just to be thorough. It's meant to be high level because a lot of the pillars, you try to build a workflow in your preclinical assays that allows you to begin to measure components of those 4 pillars. So you're kind of building the foundation for your 4 pillars. And while I didn't say it a while ago, the 4 pillars are going to be important preclinically and clinically. So your cell-based assays preclinically, your in vivo models preclinically, and whenever you go to human, when you're in clinical development in phase 1, you want to know your drug's at the right concentrations, getting to the site of action. We think we're confident it's engaging the target. It'd be great if you have an early readout on an effect on a key disease-relevant biomarker, but that sometimes doesn't come till a little later. Screening. I won't go through this here. Put a comment down at the bottom about, AI and ML is really hot right now. Virtual screening for those PIs who have a target of interest, which is structurally enabled. It's really come a long way. There's a lot of success in this space. I've experienced it firsthand. So it's starting to have an impact on drug development here and in other places in the drug development pipeline as well. Secondary assays, what is the quality of a chemical matter? It's really important to do a thorough due diligence on that chemical matter that comes out of a screen. You have to ask yourself, what are the secondary and orthogonal assays, to validate target engagement? There's, again, a lot of non-specific effects from hits that come out of a screen due to aggregation or solubility issues or polypharmacology or toxicity, whatever the case may be. So, what I think most funding groups and pharma groups, investment groups, or biotechs, they actually build these orthogonal assays where they get a hit from a screen. They'll build another orthogonal assay with a different readout to kind of help to build confidence that the effect is through engagement of your target with your hit molecule. Building the selectivity assays. You want to build cell-based assays with disease-relevant readouts. Again, it gets back to the 4 pillars. Is it getting inside the cells? Is it engaging target? Is it eliciting the desired functional pharmacology? And then is it having a relevant impact on phenotype in a cell system, whatever that may be? biomarkers of target engagement. And then early on, at least with small molecules and even some antibodies, you want to begin to start thinking about DMPK properties and some of the other physicochemical and ADME properties for your therapeutic, just so you understand your molecule before you go in vivo. And in vivo, I see a lot of PIs, they'll take 1 or 2 doses and go straight in vivo, and they haven't really properly characterized their molecule, or they don't even know if following the in vivo study, whether the drug actually got to the site of action and really engaged the target, or if the effect is specific or not. So the more you can do on the front end to be able to begin to build that case that you've got a really high quality probe, positive data from the efficacy study makes it easy to interpret these data. And investors and pharma groups and translational funding groups will know that, and they'll be able to see, we'll see what are some of the challenges with interpreting the data from in vivo studies. So you want to collect the PK data, want to have biomarkers of target engagement. Be great at this stage if you have translational disease biomarkers, i.e. those, if you've got what you think is a disease relevant mouse model or route model or whatever model you're using, if those biomarkers are the same biomarkers you could use whenever you get into clinical development. But that's not always the case. And this is probably out of scope for most academic investigators, is thinking about development of a PK-PD relationship. But I think if you can get to, if you're thinking about the 4 pillars in this context in your secondary assays and your in vivo studies, then I think it just helps build a case that to investors that this is something that's a project they should pay attention to and hopefully fund. Preclinical tractability, I touched on this really early on. Again, we encourage those who aren't familiar with thinking about different modalities to reach out to people who are. Try to find those. Early on, think about when you start in a project, what is the best modality for my project, for my target? It doesn't matter what it is, but there are lots of modalities that are available now. They're more and more becoming available every month. So it is really important to think about that early on. And there are pros and cons to each of those. And I've just picked 2 here, small molecule and a biologic. Small molecules, there can be non-specific. You can get toxicity because of the small molecule. Biologics, on the other hand, are a little more specific. So they're very different beasts, obviously, than a small molecule. And if you look down at the bottom, there are differences in probability of success between different modalities. In this case, there's about a 2-fold higher probability of success for antibodies versus small molecules. With that said, 50% of the FDA-approved drugs last year were small molecule drugs. I think it was 53%. So still, small molecules are a major player in the FDA approval, annual FDA approvals. Okay, clinical tractability. And I know in my experience, a lot of, not all, but a large percentage of academic investigators really don't think so much about clinical tractability. And this is extremely important because the funding groups and investors will be thinking about how long it's going to take me to get an answer in the clinic. What's it going to be the cost? How many patients will it take? How am I going to find those patients? And you can read at the top left and work your way down. Do your due diligence on the right indication. And that obviously has a commercial side as well to think about. If it's a rare disease, are there expansion indications? What is the size of the treatable patient population? I run across proposal after proposal where, pick Alzheimer's, for example, where the size of the AD patient population is enormous. But if you look at their mechanism of action, I think you'll realize that they can't treat all those patients just because of the disease and their mechanism of action. So I think it's really important to think about the size of your treatable patient population, because that's where the commercial side will come into play. Who are your responders? How do you find those people who are going to be your responders, whether it's genetic data, whether it's biomarker-driven patient selection, figuring out early on, how do you get an early read on patients who are going to be responders to your therapeutic, and how do you build a clinical development program to include those patients? I think will decrease your cost and help hopefully decrease your timelines. I think if you've got to do a 5-year cardiovascular outcome trial or 10-year cardiovascular outcome trial versus a 6-month or 1-year clinical development trial, it's much more digestible from a cost perspective and timelines perspective if you can obviously get an early read sooner rather than later. You need obviously the robust biomarkers. It's really important to understand what are the FDA guidelines around and the requirements for FDA approval for your lead indication. And then down at the bottom, there's no perfect project. There are always risks. It's important to put into context, does the unmet need for the proposed indication justify the risk that are associated with the approach? And there, again, it's if you think about cardiovascular disease versus oncology, they're very, very, very different profiles that you would look for in a molecule from an efficacy and safety perspective. Unmet need in the medicine proposition here, again, at the top, treatable patient population. You look at incidence, prevalence. Is there an opportunity for orphan designation? Disease burden. What are the regulatory requirements? And on the right-hand side, what are the alternative treatments? How many are there? Are they available? How is your approach going to be different from these alternative treatments? And is there any remaining comorbidity that you can actually address with your approach? Bryan, you want to take this?
Thank you, Jon. Before I jump in, most of my experience has been in the academic world, so I am directing most of this based on my experiences here. But I would say that a lot of what you've seen thus far and what you'll see thereafter, the context to put it in is that you don't need to be an expert in all these things. These are, as a matter of fact, I had Jon in. He actually helped me from a consultant standpoint about a week ago with a couple of PIs that had small molecule endeavors in early stages. And, an hour of Jon's time was immensely valuable, and it's not a whole lot. So the thing is to think about getting to the right experts earlier. And I think that's one message I would like to leave behind. So jumping into commercial considerations, the first one I have is path to intellectual property. What I'm saying is you may not have IP already, but be thinking about intellectual property because you need to build it. It's a part of what's going to attract outside investors. And so you want to think about building intellectual property that's valuable. The other thing that I found that PIs aren't as in tune to are the next couple of bullet points around competitive landscape and differentiation. You have to have a competitive landscape to include a target view as well as indication, and think about marketed and pre-market pipeline. But please bear in mind, the market changes even daily, and you have to incorporate the new additions or subtractions or new standards of care approaches. So don't think that what you did 2 years ago also is the same today. So you always have to stay current with that. Differentiation is obviously very important because to get anyone's attention on the investor side of things, you're going to need to be able to show that you can that there's a valid reason to integrate your product into the existing products. Current market feedback. What we do a lot here at Duke is when we have once I've gone out and I've done some initial screening of projects, applying some of the rationale that Jon talked about earlier. I mean, is it genetically validated? Is it human validated target? Then looking at top level kinds of things. What's going on in the marketplace? Who else is chasing this target, et cetera, et cetera. Then it's kind of nice to build a straw man sort of a presentation to get in front of the VC community, and we call them friendly VCs, folks that we trust. It's just early days. Give us your read kind of thing. What comes out of it is a lot of good feedback. And if you embrace that feedback and scrutiny, it helps you build your project moving forward. So it's really important. And also, when you're going to these folks, you can count on their view being current. So if you're missing something, they certainly know it most of the times. And then the last bullet point I have is value de-risking profile. This is just so you understand how people think that are in the investment world. They're looking to increase value over time. What they want to understand is what are the de-risking experiments that are usually the toughest, that might kill your project, because those are the ones that increase the value the most to them. Because usually those things that they bring up, they're the ones that kill the project, or the doubts pervade the investment community, and you're trying to overcome that one. So don't shirk those. You might have to find the best experiments to just attack those head on, and understand that when you get to the other side with success, you have a much better chance of finding money. Thank you, John. If you move to the next. So, target product profile. You're familiar with these. John mentioned earlier. Here's one populated. The main points of this slide is just to remind you that these are guardrails. Actually, the guardrails mainly on the left. I mean, if you breach any of number one, this TPP takes into account the financial thesis that an investor would look at. So by the time you do this, there's probably investors interested. They might be helping you pull it together. You've got a spin out, and you've got a CEO, and they're trying to build this. But bottom line is, this is set up so that it's a reference point to always be looked at. It can be modified, but the point is, and if you take something like dosage regimen, let's look at dose regimen, where there actually is a split. I mean, the ideal on the right is 1 to 2 times per day for acute pain, and then also weekly, monthly for chronic. So that's really the wish list. But at a minimum, it has to hit the daily or weekly or monthly for chronic. And if one is not going to reach that, then one has to go back and reassess and add that change into your financial planning, because in the world of finance, they may say this is a no-go. So that's why this is there. That discipline is there for a reason. John, I think you got the next slide.
Yeah, I do. Thanks, Bryan. Yeah, and this is, we don't have a lot of time because we need some time for Q&A, but I wanted to put this in here which is your target development candidate profile. The TPP is for your product. The TCP actually is the profile of the therapeutic to deliver the TPP. And here are 2 examples, small molecule example and a monoclonal antibody example. And this is where, all of the assays you built in the preclinical execution phase of a project, you're populating these tables. And then ultimately this will end up in an IND document. But I think, for the purposes for the academic investigators, they usually, and I don't think it's really critical that, early on in the academic setting, you just don't have the capabilities to populate this entire table. You're going to have to go somewhere else and get to translational funding. But you can at least start to think about, okay, what am I going to need to achieve? What is my ultimate development candidate profile? And development candidate is the molecule that goes into IND enabling studies and beyond, assuming everything looks okay. So that's the difference between a TCP and a TPP. On the right-hand side, it's monoclonal antibody. I won't go into all the details there, but it's just something to think about, with the requirements, the profile of your particular molecule or your antibody or whatever the case may be to actually deliver the TPP, and what does it need to have to be successful. All right, Bryan.
All right, so now we're getting to the end of the presentation, and it really comes down to, where do you find the funding? Everybody's heard about the valley of death. That's a cliche, but usually cliches are true, I've found. And the point of this slide is just to remind you that in the world of government funding, the thinking changes from that as you move into industry. And I know that a lot of the PIs that are on this call, they know how to please the funders. You know who you're talking to and what they're asking for, and you craft your grant to answer those questions and to have success. You'll have to do the same thing as you're moving towards industry. But the view of that changes, and the main point is that you're building something when you're building out what you're doing in the academic world. But when you start moving into industry, you're actually you're looking for no-goes to rise to the top, and you're looking for killer experiments. This may be not something that you're thinking about doing as much, but that's what's going to be required to convince the folks with this kind of money that they're going to jump in. So you may as well go embrace it. Next slide. So this is by no means an expansive list. It's just an idea. I think that the message to take from this is all of these allow you to retain your IP rights within a university. They're all mainly designed to be run inside a university and to help you continue to build value in your asset before you spin out. The CARB-X also allows for companies to come in. But the point is, you need to stack up some, these are what I can view as translational vehicles. You're obviously familiar with catalyze, but most of these things aren't going to get you all the way there. So all these things do is get you really primed to go after money that is more what we call dilutive, where investors are going to start coming in. And so the ones that I have at the bottom are others that are also to be considered. SBIR and STTRs would require you to have a company. So the point of this is, I can't go through this slide, is to figure out what variables matter the most and understand that any one funding vehicle may not be the only way you move this thing forward. You may have to stack more than one together, and you may have to also understand that the funding intervals are such that you're going to have to be able to leapfrog from one to the other. Thank you, John. You can go to the next. Lastly, a lot of the hallmarks of these is most of these that I had on the prior slide and that I'm running into are requiring hits for therapeutics, a hit at a minimum. Additional characterization is nice. Somewhere in that hit to lead is a nice soft spot, and they'll take it later, of course. End product visualization is important. You need to at least have a pretty good sense for what your therapeutic hypothesis is, and you're able to explain it and be able to explain how you would fit in to that and actually compete. High level competitive landscape and differentiation thesis. Make sure your IP is unencumbered. So they want to know it's not optioned. They want to know that they don't have to deal with 2 or 3 other institutions. And if they do, they want to know that an interinstitutional agreement is already in place. So make sure you got your IP shored up if you have IP. Having a framework for a development plan moving forward, some ideas about what you would do to de-risk this is important. And most of these funding agencies can help you get into the granular nuts and bolts if you get funded. So that's what I found with most of these. That's all I've got. Thank you.
I think that's it for the presentation. We hope that there's some nuggets in there for academic investigators as well as people that may be in early stage biotech space. Yeah. So again, we thank everyone for joining today. And Patrick, I think you were going to get the questions together and see if there are any from the audience.
Yep, we do. Thank you, Jon and Bryan, both for that presentation. We do have a few questions. We have about 10 minutes left in the session. So if you have any additional questions, please put those in the Q&A window. The first question here, does the new in vitro human model system improve or speed up the drug development process?
I think it's both. I think it does. I think it improves up. I mean, it just, to me, those systems actually, help you strengthen your case for the therapeutic hypothesis. And I think ultimately, if that proves translational, then it will ultimately have an impact on your timelines and cost as well. I think that answers that question. I hope. If not, let me know.
Okay, next question. What is the business model of Pinnacle Hill?
Yeah, that's a good question. That is a partnership between Deerfield Healthcare Investment in New York City as well as UNC Chapel Hill. Bryan, he manages Duke's. It's a similar thing to Duke, 4 Points Innovation. It's basically Deerfield's mechanism of trying to tap into innovation in academia. And there are certain rules of engagement. Deerfield has a very robust strategy. Projects that align with that particular strategy. This is non-exclusive. Projects that look interesting on the institutional side can be evaluated by Deerfield, and then those that look attractive and everything lines up in terms of the PI's interest And the criteria met, everyone on the Deerfield side, the investors, their drug development team are aligned, and there's a lot of financial support that goes behind that. But it's not. I mean, it's projects that are fully vetted and pass all the meet all the criteria that Deerfield are looking for to move into a full drug development program. I hope that answers the question. I obviously can't get into all the financials or any of that stuff. Bryan, do you have anything? I mean, for 4
No,
points.
I thought that was well said. So I don't. There's the same. The main thing to also mention too is that it's an academic consortium. So Duke's a part of that, and our arm is called 4 Points, and there are 30 universities involved in this. So
They're very good. They're very, very good. They're great to work with. They got a strong drug development team. They got the investor side as well, and they're very, very strong team. So I enjoy working with them.
Okay, next question. Why do half of all candidates fail after clinical trials? Does this assume favorable safety and efficacy data in clinical trials that nevertheless do not obtain approval?
Well, if I understand the question, for me, I think it's early on, years ago, people didn't pay attention. 10 years ago, people did not pay attention to human translational data at all, and they used mouse models or rat models to actually predict efficacy in a human. And I think what we've learned over time is that those models are not predictive of human biology. So I think that's one of the reasons. A lot of these, there aren't very many preclinical models that are translation. I mean, there are some, but that's probably an overgeneralization. But I think, the shift, the cost of doing human genome sequencing has gone down. That's allowed companies to generate just loads and loads of human genetic data where they can look at genotype-phenotype relationships and associations with disease. And so I think if you're looking at data where you've got gene of interest, a SNP in a gene, and you see a phenotype or marker or impact on disease outcome, I think that just de-risks the biology, which again, as I said earlier, that's 70% of the cost and 60% of the time. So they're trying to move the needle in terms of decrease their costs, improve their efficiencies, and early on try to understand whether your gene of interest could be relevant in a human setting. I hope if that doesn't answer the question, please, come back.
Thank you, Jon. We do have a couple more questions, but I did want to point out that the Catalyze feedback poll is now open. If attendees wouldn't mind taking a few moments to fill that out. I also wanted to draw attention to some information on the screen. Our next Catalyze webinar will be August 28th, which will cover somatic cell genome editing and complemental animal research and experimentation. We do have a couple more questions here, Jon and Bryan. Maya says this talk was very helpful. My lab is working on a clinical decision support AI algorithm that's taken a drug development approach over the past 15 years, and we're developing a commercialization plan. All of the info that you've shared is very much aligned. Do you have experience with these processes for clinically impactful AI with evidence of mechanism of action developed with either federal funding, K or R01 type grants?
Bryan,
I do not.
You don't? Is that what you said?
I don't, Jon.
Yeah, I don't either. I mean, most of my experience with AI is usually in preclinical development. I know, there's a lot of emphasis on using AI and ML in the imaging space. Beyond that, that's outside, to be honest with you, that's outside the scope of my expertise. It sounds like it's Bryan's as well.
Yeah, what I will say is if this person's a Duke PI, they should just come to our office at OTC, because there is experience within the group, but it's not something I handle on a regular basis. So, I mean, we get a lot of AI technologies across the scope, but I haven't heard of that one particular Yeah, I'm not, I haven't seen one that looks like that. I do actually see some of these. But I haven't seen one that addresses that problem. So it sounds like a good one to try to expand and use. And if
Okay,
you're at another university, then go to your other university's tech transfer office. Hopefully they can help you.
okay. Next question. Are the UNC and Duke programs represented here today providing similar support for cell therapeutics translation?
Yes, I can say yes at Duke. I mean, I was just in a meeting with a PI, hour and a half meeting before this at cell therapy.
Yeah, I think UNC is really strong in cell therapy, CAR T in particular. That and they've even gone so far as to build, a GMP facility for manufacturing CAR T therapies in oncology, and some of the clinical trials are run at UNC. So yeah, they've invested in that space, like Bryan and at Duke.
Okay, and looks like we have time for maybe one or two final questions. How are funders thinking about drug delivery approaches, such as nanomedicines that propose to take a validated drug and improve safety and efficacy? What are common issues or things to de-risk here?
I mean, I'll comment, John, if
Please.
you'd like, and then you can add. I mean, I think what you got to do is you got to really choose the right drug, right? You've got the validation target piece is not an issue like John mentioned, but the differentiation piece is. So I think investors are going to be interested in going after therapeutics where there's some serious flaws, and there's some way to overcome those flaws with the technology that you have. And don't fool yourself on the flaws. I've had PIs talk to me about flaws of certain standards of care, and then when I really start digging in and we start going to other third-party references, it's not as big of a concern, or they don't want to go through the trouble of circumventing the insurance payers to get something else on board that they think might be more money, et cetera, et cetera. So all I'm saying is, when you take this one, you're threading a needle. I think there's a lot of value there, but I think you need to make sure that you've done a lot of market assessment and you've chosen the right one to go after first, or maybe the top three with reasons why, and make sure that you do some customer discovery and talk to other physicians and make sure that you're getting varied opinions and that you're hearing the same thing over and over again. I hope that helps. John, you have anything to add?
No. That's probably more your space than mine.
So thank you, Jon and Bryan, for your time this afternoon. That does bring us to the top of the hour. There are a couple questions that we didn't get to, but we will save those and perhaps we can answer those offline. But once again, I draw your attention to upcoming NHLBI events on your screen. And I'd like to thank everyone for attending. And once again, thanks, Jon and Bryan. And I hope everyone enjoys the rest of the afternoon. Thank you.
Thank you.
Thank you. Bye-bye. Have a great day.